Welcome to the first of a series of videos where we look at acid-base balance. In this introductory video, we're going to talk about the difference between acid and base. We're going to define the body's normal pH and why it keeps its pH at that level. And we're going to start looking at buffers in which the body is going to use to control this pH. First thing we should start with is the definition of an acid and a base. An acid is a compound that can freely donate hydrogen ions to solution. When we're talking about pH, what we're really talking about is the concentration of hydrogen ions in solution. As we increase the amount of hydrogen ions in solution, we decrease pH. When we decrease the number of hydrogen ions in solution, we increase pH. What we call that is an inverse relationship, or our pH is inversely proportional to the amount of hydrogen ions that we have in solution. So an acid, when we say, is something that can donate a hydrogen ion, means that it has a hydrogen ion bound to the compound, which it can donate to solution. So as the hydrogen ion dissociates from this compound, becomes free in solution, what we're going to see is an increase in hydrogen ion concentration in solution, which subsequently, because we have an inverse relationship, is going to decrease pH. So acid is a compound that has a hydrogen ion attached to it, which can dissociate or can give away that hydrogen ion. It gives away that hydrogen ion and becomes part of the solution. And as that solution's hydrogen ion concentration increases, we start to see a decrease in pH. A base is the opposite of this. So a base is a substance that will accept, or a compound that will accept, a hydrogen ion. So we have a compound that generally will have a negative charge that can accept a hydrogen ion out of solution. So what happens is this hydrogen ion leaves the solution, binds to this compound, and as a result, we see a decrease in hydrogen ion concentration in that solution because that hydrogen ion is now bound to this compound. So as a result, we see a decrease in hydrogen ion concentration, which is going to lead to an increase in pH, which is going to make the person more basic. So an acid is a proton donor, which is going to increase hydrogen ion concentration in solution, decrease pH, and make someone more acidic. A base is a hydrogen ion acceptor, or that hydrogen ion is going to leave solution, bind to our base, so we'll start to see it living on this base. And as a result, we're going to decrease hydrogen ion concentration, and we're going to increase pH, and someone is going to become more basic. So the body lives in a constant balance between acids and bases, and it's going to use buffer systems to do that. And the body uses those buffer systems to keep body pH in a very specific range. Generally, when we're talking about the homeostatic pH, or where the body keeps pH in homeostasis, we're talking about arterial blood. And the core values for what that pH is going to be is 7.35 to 7.45. So normal arterial blood is going to have a pH of 7.35 to 7.45. And the reason for this is because it's the homeostatic safe zone for our proteins. So as we know proteins make up a large amount of our body's ecosystem, and those proteins are going to help us survive. When we're looking at survival of those proteins themselves, they need to live in a very specific pH. So if our pH is between 7.35 and 7.45, those proteins are folded up nicely and they can do their job. It's our protein. The problem is, as we leave this safe zone, so pH starts to fall below 7.35 or go above 7.45, that protein starts to unwind and unfold, and we get what we call protein denaturation. As a result, those proteins are no longer going to be able to function. So pH is required to stay between 7.35 and 7.45, because if we move out of that safe zone, these proteins can no longer function. They unfold, denature, and now we're going to see the person most likely die. So arterial pH sits between 7.35 and 7.45 in order to maintain adequate protein folding. Some differences lie in different fluids in the body. Venous blood, for example, has a slightly lower pH. It sits between 7.32 and 7.42. Our intracellular fluid and our intracerebral fluid also have a lower pH. A lot of this is due to an increase in CO2 levels in these solutions. But mainly what we're looking at is arterial pH, and the values that we're most concerned about is that 7.35 to 7.45. So in order to maintain this safe zone, or to keep someone in this range and not fall below these levels, the body is going to use buffers. And there's three core buffer systems, or ways in which the body can buffer for changes in acidity or alkalinity. So the three different types of buffers that we have are chemical or protein buffers. And those are our fast-acting buffers. So chemical or protein. And these buffers, like I said, are fast-acting, and they respond to small changes in acid-base balance. So these can fine-tune acid-base balance, but they're not very good at responding to large-scale changes in acid-base balance. So they're fast-acting and respond to small changes. The other system that we see aiding in acid-base balance is our respiratory system. So the respiratory system plays a role in acid-base balance. It is slower than the chemical buffer system, but it's not as slow as the third buffer system that we use, which is the renal system. The renal system is going to respond to large changes in acid-base balance, but it's going to do so more slowly. So the renal system is large changes, or responds to large acid-base changes, but it's going to do so much more slowly. In this video, we're primarily going to focus on our chemical or protein buffer systems, and we're going to take a look at the different chemical and protein buffer systems that are available to the body. This buffer system is the most important buffer system, is because what it houses the bicarbonate buffer system. So we're talking about the bicarbonate buffer system. We're looking at the primary way in which the body is going to balance acids and bases. So this is the bicarbonate buffer system. And the bicarbonate buffer system is characterized by an equation that can shift in both directions. If you know anything about chemistry, it functions on what we call Le Chatelier's principle, or basically it's a way in which a chemical reaction is going to maintain homeostasis. So this equation is written like this. We have H2O plus CO2. When we're talking about this in terms of the body, we're looking at body water plus CO2, and generally that CO2 is being produced by the breakdown of sugars and cellular metabolism. Now, the next step in this equation is H2O and CO2 can be combined to form H2CO3, which is carbonic acid. And then the third phase of this equation is that H2CO3 can be broken down into hydrogen ions and bicarbonate, which is written as HCO3 negative. If you look at the difference between these two things, where we're looking at a buffer system, a buffer system is basically made up of an acid and a base. We can use the acid to donate hydrogen ions. We can use the base to accept hydrogen ions, and this falls exactly into that category. Carbonic acid can donate a hydrogen ion in order to make solution more acidic. HCO3 negative can accept that hydrogen ion to take hydrogen out of solution and make it more basic. And that's the general principle of the bicarbonate buffer system. We can shift to the right or shift to the left in order to produce more hydrogen ions, or take hydrogen ions out of solution to make that solution more basic. So when we're looking at the way we can shift from stage 1 to stage 2 to stage 3 of this equation goes both ways. So it depends on the concentration of these elements of the solution. So for example, if we start to see a rise in hydrogen ions, or we start to see an increase in acidity, what the body's going to do is bind to this bicarbonate, make H2CO3, which subsequently turns into H2O and CO2. Whereas if maybe we stop breathing so well and we start to see way too much CO2, we can alternatively shift to the right, where we see a production of carbonic acid. We shift further to the right, where we see a production of H plus and HCO3 negative. So The important thing to remember about this equation is it's always shifting back and forth in order to maintain homeostasis. So if I get too much of something on this side of the equation, or I start to see a huge buildup of things over here, what we're going to see is a shift to the right to try and minimize that or to equalize that. Conversely, if I start to see a shift or I start to see a huge buildup of things on this side, we're going to shift that back over to the left in order to accommodate for that. So the bicarbonate buffer system is made up of H2O and CO2, which shifts into H2CO3, which is carbonic acid. There's an enzyme that works here, which is called carbonic anhydrase, that's going to facilitate that reaction. And then H2CO3 can break down further into H plus, HCO3 negative, and alternatively that can shift in the alternative direction or the other direction as well. Now the one thing that's important to note is that the ratio of bicarbonate ions to carbonic acid ions isn't 1-to-1. So in order for this buffer system to work adequately or to actually favor buffering, we generally have to have more HCO3 than we have carbonic acid. And the reason for that is if we're thinking about a solution. So we have, say, a beaker here that's full of solution. So we have our solution. It's got some H2CO3 in it, which we know is going to give off hydrogen ions. So we'll make these little pink guys our H2CO3, which are giving off hydrogen ions. Those are hydrogen ions. And then we have bicarbonate that's living in here as well. So we've got bicarb floating in this solution. Now, according to this equation, what should happen is if we start to see more hydrogen ions than there's bicarbonate, or we start to see an increase in hydrogen ion concentration, we should start seeing a combination of this bicarbonate to hydrogen ions and shift this equation to the left. Now, the only thing that's different, or the only thing that we need to remember about this, is in order for us to actually have an equilibrium, or for this equation to actually work, or buffering to work, we need to have a ratio of bicarbonate that's much higher than our carbonic acid. So for every one carbonic acid, in order to make this scale balanced, for every one carbonic acid that we actually have 20. So there's our one carbonic acid, we have 20 H2CO3. So every one bicarbonate, or for every one carbonic acid, we have 20 HCO3 negatives, or bicarbonate ions. So for every carbonic acid, we have 20 bicarbonate ions, or we should have 20 bicarbonate ions. And the reason for that is that not every one of these bicarbonate ions is going to pick up that hydrogen and turn into carbonic acid. So if we oversaturate, or we put many more bicarbonate ions in this solution, our likelihood of picking up that hydrogen and shifting that equation to the left is much more likely. So we create a solution, the body creates an environment that favors buffering. So the only thing, or the takeaway points from this, is that this equation will move in either direction. So it can move to the right or move to the left in order to balance changes in acidity or pH. And in order to do that, the body maintains a ratio of 20 bicarbonate for one carbonic acid. And that's going to allow or promote buffering. If you think about the importance of that, is if someone starts losing HCO3 or starts losing bicarbonate, that starts to make a big difference, or it starts to have big changes and consequences in our acid-base balance, because we need that bicarbonate concentration to be quite high in order to maintain acid-base balance. And this will make a little bit more sense when we talk about the renal system and why it's so important that our kidneys function properly to reabsorb bicarbonate ions. So again, if we just look at the takeaway, or take a look at the takeaway points from this, is one, we need to remember that this system can go in either direction. So the bicarbonate buffer system can shift to the left or right depending on pH. So it can shift to the left or right depending on pH, depending on the concentration of CO2 or hydrogen ions. So as we start to see more of these ions building up, or more of these compounds building up on this side, we can shift to the right. If we have more of these compounds building up on this side, we can shift to the left. And the other thing to remember is that we have a 20-to-1 ratio of HCO3 to carbonic acid. And the reason for this is to favor buffering. So just the main thing here is that this promotes buffering. So that's the primary buffer system that the body is going to use. That's our bicarbonate buffer system. This equation is going to show up when we talk about respiratory buffers. It's going to show up again when we talk about renal buffers. And it's the primary way in which the body is going to maintain acid-base balance. It works quickly, and it works better when we're looking at small changes in acid-base balance. If it's not a small change, then we need to start using things like the respiratory system and the renal system to manage those alternatives in pH. The other type of acid-base balance, or the other type of buffer that we're going to talk about in this video, is protein buffers. So when we're looking at protein buffers, we're looking at using the proteins within the blood in the body to manage pH. So when we're looking at proteins are compounds that have two different groups on them. So a protein is going to have a amine group, so an NH2 group, and a protein's also going to have a carboxyl group, which we'll demonstrate here as OH. And this either side of this protein can act as either a acid or a base, which will allow us to actually buffer. So the amine side can actually accept hydrogen ions. So if we're looking at the amine side of our protein, what can happen is as our body's pH starts to decrease, or say we start to become acidic, so if we say our normal amount of hydrogen ions is 2, now we see 3 ending up in solution. What can happen is this amine group can accept a hydrogen ion. So this hydrogen ion can be removed from solution, can bind to this amine group, and now instead of having NH2, we have NH3. So we removed a hydrogen ion from solution. It's now bound to that amine group, and it's helped buffer the solution. So we took something that was acidic, and we've moved towards homeostasis. Now the alternative can happen when we're looking at the carboxyl group of our protein. We can use that carboxyl group to donate a hydrogen ion. So we have NH2, we have our OH, and what can happen is we can have that hydrogen ion dissociate from the protein and make its way into solution. So if we said our normal hydrogen ion concentration is 2, if we only have 1, we know we need another one in our solution, or we need to, we have a basic solution that we need to make more acidic. So what we can do is we can take this hydrogen ion, remove it, put it into solution, and now again, we've moved back towards our homeostasis. So our protein buffers use hydrogen ions, or can accept or give hydrogen ions in order to maintain homeostasis. So we're looking at proteins. We have an amine group that can accept a hydrogen ion that's going to make the compound more basic, or it's going to take acid out of the compound. Or we have the alternative, which is the carboxyl group, which can give a hydrogen ion and decrease the pH. All right, the last buffer we're going to talk about, or the last buffer we're looking at in the chemical or protein buffer system, is our hemoglobin buffer system. So just as we can use hemoglobin to carry oxygen, we can actually use hemoglobin to bind to hydrogen ions. And as a result, because we can bind that hemoglobin to hydrogen ions, we can use it as a buffering system. So the last one we're going to look at is our hemoglobin buffer system. And in order to look at that, we're going to take a look at a red blood cell and then our cells themselves. So as we perform cellular metabolism, or as we're having cellular metabolism, we know we're accepting oxygen and we're producing CO2. So we're looking at the cells that we've delivered oxygen to, and they're going to be releasing CO2. So the red blood cell that has come to these cells and delivered a hemoglobin with oxygen bound to it can now do something else. So what we do is we take the CO2 that has been produced by cellular metabolism and it's going to move into our red blood cell. This little red guy here is our red blood cell. So we know that our bicarbonate buffer system uses CO2 and H2O to transition through that equation. So that CO2 will bind to H2O in our red blood cell. It's going to form carbonic acid, H2CO3, which is then going to form hydrogen ions and HCO3 negative, or bicarb. The good thing about having this bicarb or moving this equation to the right and producing hydrogen and bicarb is that we know we have to have a 20-to-1 ratio of carbonic acid to bicarbonate. So if we can do something to get rid of this hydrogen ion, we've now created a free bicarbonate, and that's going to help us maintain that ratio, or that high ratio of bicarbonate ions to carbonic acid. So what's going to end up happening to this hydrogen ion is it's going to bind to a hemoglobin. So that hemoglobin molecule initially came to the cells with an oxygen attached to it. So what happened is the oxygen is going to leave the hemoglobin. It's going to go out to the tissues to supply the oxygen it had, and that hemoglobin is going to be free. What's going to happen is that this hydrogen ion is going to come down and bind to our hemoglobin molecule. So as we shift over, we now see a hemoglobin bound to hydrogen, and we've pulled that hydrogen out of solution. So we created a hydrogen ion, we pulled it out of solution, and now we've adequately buffered that acid that's been produced. Now we still have a HCO3 remaining, and what we're going to see here is what we call the chloride shift, because we want this HCO3 to be in our arterial blood to help with buffering. So what happens is we take that HCO3 and we move it out into the bloodstream. So we see HCO3 negative moving out to the bloodstream. Now we have to exchange a molecule and a charge. So the best way to do this is to exchange chloride, or a chloride ion, out of the blood into the red blood cell, and this is what we call the chloride shift. So chloride moves into the red blood cell, exchanges itself for bicarb, and now we have bicarb out in solution where we can have that buildup of bicarb where we can maintain this 20-to-1 ratio so that we can adequately promote buffering. So the last thing we're talking about here again is the hemoglobin buffer system. This is where when hemoglobin comes to the tissues, it's offloaded its oxygen. What we can do with the CO2 that's being produced by cellular metabolism, let's run it through this bicarbonate buffer system. I start to see a buildup of CO2, so I start shifting this system over to the right, which is going to produce hydrogen ions and HCO3. I take that hydrogen ion, I bind it to my hemoglobin, pull it out of solution so that I'm not becoming too acidic. I take that bicarbonate ion, I push it out into the bloodstream by exchanging it for a chloride ion so that I still maintain my charge that I need to maintain across that membrane. And that HCO3 can now be free in the arterial blood, and it's going to help create this 20-to-1 ratio of bicarb to carbonic anhydrate, or carbonic acid, that we said is important to actually promote buffering.